Hello everyone, today we are going to learn about plant cell reproduction. The growth and reproduction of individual plants are mainly the result of the increase in the number, volume, and functional differentiation of cells in plants. The increase in the number of cells, or reproduction, occurs through cell division. Today we will look at the reproduction of plant cells in terms of the three ways of cell division: mitosis, meiosis, and amitosis. First, let's look at mitosis. Mitosis is also called indirect division. It is the most common and widespread form of cell division in plants. During division, chromosomes and spindles appear, hence the name. After mitosis, a mother cell produces two daughter cells with the same number of chromosomes as the mother cell. The process of mitosis is relatively complex, including two stages of nuclear division and cytoplasmic division. Mitosis is a continuous process. For the convenience of study and description, the entire process is divided into four stages: prophase, metaphase, anaphase, and telophase. Let's start with prophase. As shown in the figure, the chromatin filaments in the early nucleus condense into chromosomes, the nucleolus disintegrates, the nuclear membrane breaks, and spindle fibers appear at the two poles of the cell. Then comes metaphase, when chromosomes align on the equatorial plane, the center of the cell, and the spindle is fully formed. This period is convenient for counting chromosome numbers. Next comes the anaphase, in which the chromatids of individual chromosomes separate and move from the equatorial plane to the poles of the cell under the traction of the spindle filament. Finally, in telophase, the daughter chromosomes that reach the poles become chromatin filaments, and new nuclear membranes and nucleolus appear, forming two daughter nuclei. At the same time, the cytoplasm divides, and the spindle filaments on the equatorial plane gradually shrink and thicken, forming the phragmoplast. The vesicles separated from the Golgi apparatus and endoplasmic reticulum converge on the equatorial plane and fuse with the microtubules of the phragmoplast to form the cell plate. The cell plate gradually expands centrifugally until it makes contact with the wall of the mother cell, completing cytoplasmic division and forming two new daughter cells. So that's what happens in mitosis. Now, let's look at meiosis. Let's look at the similarities and differences between them. Meiosis is a type of cell division that occurs during sexual reproduction in plants, producing gametes. Meiosis is a special kind of mitosis. The whole process consists of two consecutive divisions, but DNA replicates only once. After meiosis, a mother cell yields four daughter cells. The number of chromosomes in each daughter cell is reduced by half than the mother cell. So it's called meiosis. Now, let's look at the process. First, there is the first division, which, like mitosis, is divided into four stages: prophase, metaphase, anaphase, and telophase. In prophase I, homologous chromosomes pair up, a phenomenon called synapsis. Crossing over occurs between chromatids, and exchange and recombination occurs. This phenomenon is called crossing over. Finally, chromosomes shorten, the nuclear envelope and nucleolus disappear, and spindle fibers appear at the poles. Then comes metaphase I, similar to mitosis, where pairs of homologous chromosomes are arranged on the equatorial plane in the middle of the cell, and the spindle forms. Then, in anaphase, under the traction of the spindle filament, the two chromosomes in the homologous pair separate and move toward the poles, respectively. Thus, the number of chromosomes at each pole is reduced by half, which is different from mitosis. Finally, there is telophase, when the chromosomes reach the poles and a nuclear membrane forms and a nuclear envelope forms around each set, resulting in two daughter nuclei. The first meiotic division ends, followed by the second division. The second division is essentially a standard mitosis, also divided into prophase, metaphase, anaphase, and telophase, as shown in the figure on the right, and finally forms four daughter cells, which will not be repeated here. After meiosis, a mother cell forms four daughter cells, which further develop into eggs or sperm. There are only N chromosomes. When sperm and egg fuse, the resulting zygote has two N chromosomes, and the offspring developed from the zygote have the same number of chromosomes as the parent, ensuring the stability of the number of chromosomes of the species. During this process, genetic exchange occurs, providing potential for biological variation. This diagram simply shows the process of mitosis and meiosis, and the difference between the two can be seen. Meiosis consists of two successive divisions, forming four daughter cells with half the number of chromosomes of the mother cell, and each daughter cell is genetically different. Mitosis divides only once, yielding two daughter cells with the same number of chromosomes as the mother cell. And finally, amitosis. Amitosis is a simple form of division in which the interphase nucleus divides directly without undergoing the stages of mitosis, resulting in two roughly equal daughter cells. It gets its name from the absence of a spindle filament during division. Based on nuclear morphological changes, it can be classified into types such as transverse constriction and budding. The process of amitosis is simple, the division speed is fast, and it can meet the needs of rapid reproduction of plant cells. That's all for today's lesson, and I'll see you next time.